mathematical theories and stochastics with experiments and experimental data. We'll now have a presentation by Christof Schuette from the Free University of Berlin, who will be describing another important aspect of engagement of stochastic mathematical research in connecting with science, which has to do with using it to interpret data that's produced from simulations that are generated to try to understand biological data. So it's my pleasure to introduce Christof.
Thank you very much, Pete, for the introduction and for the kind invitation. Originally, I wanted to talk about data and reaction diffusion systems, particle dynamics, but this morning so many things were introduced so nicely that I skipped half of the talk and replaced it by something else. So the first half of the talk will be about something which perhaps is needed to understand the background of what I want to do in the last part of the talk. So there will be a longer introduction in a certain sense now. The work I'm going to present, especially in the last part, is with Steffi Winkelmann, who is sitting in the audience there. So if you have questions, you can also ask her. Let me start with molecules instead of cellular things. Let us imagine we have a protein. That's really a collection of atoms, and we have bonds between them. The bonds are forces between these atoms and keep everything together. Assume that we know the nature of these forces, which is today true to a certain extent. And then there is a background of something like this protein, and then, but normally it's only looked or visualized like a kind of secondary element structure. In this case, a sandwich of 3 beta planes or beta sheets, and then you can ask, okay, that's perhaps the folded structure of the protein, but the protein is not something which is not moving, it's doing everything all the time, so how does it fold into the structure? And then there's this movie, it's already a little old now, almost 10 years, by the Schulte group. At that time it was really state of the art, and they started from an unfolded structure and then did a very long computer simulation, the longest computer simulation they could do at that time. And I wanted to understand the folded structure, and especially the time, the rates of going into the folded structure. And if you look at the movie, then you see everything is happening, but you never find the folded structure within the simulation. So what is the case still in this particle-based simulations in molecular dynamics, one of the key problems is that there are so many timescales that you want to more or less involve in your computation, that even on the largest computers, you cannot really go to the timescales you're really interested in. So we have to start here. The time steps are somewhere here, because you have so many atoms in your system, so many particles. And even to go here, you would have to cross 9 orders of magnitude for, let's say, 100,000 atoms, and that's problematic even nowadays. So nowadays, perhaps we can go there with special purpose architectures there, and then you can do something else and perhaps go a little longer. But still, really interesting schemes at the moment you cannot access. So especially if you want to have rates, so not only one event of folding a protein, but the rate of folding, for which you would need statistics, it is still infeasible to compute the most things by conventional MD. And so that's a situation in which this field, so this particle-based, atom-based computations in molecular dynamics, that's the big challenge there at the moment. And what I want to say is that if we want to do cellular things, so the motion of lots of proteins in a cell and their reactions between each other, and then everything else, mRNA and whatever, then we are in a similar situation, because if we extend our timescales to the timescales where we are interested in let's say, cellular processes, then again we are in a similar situation. The time steps that we are allowed to do are very small compared to the processes we are interested in. So again, the same problem appears. How can we solve something like this? How can we compute quantities knowing that we are not able to do it all in all details? So that's in the biology, or especially in the biophysics discussion, is called coarse graining. So how can we coarse grain time and space in order to be able to do something like this? And as a first step, I want to explain you what has been done in the molecular dynamics sector. So this particular kind of stochastical particle systems, how has it been dealt with in the last 10 years there? And then I want to explain you how perhaps we can deal with the same question if we are dealing with cellular processes. So this is a snapshot of molecular dynamics for a very simple molecule. And what you see is that all over rotation and translation has been skipped, the water around it also, and you just see the molecule itself. And if you look at it, then you see it's vibrating and oscillating around. But if you take a different view of that, then you see that there are some sets in which the dynamics is running for some time before it jumps to other sets. And inside of this set, the dynamics stays for a certain period of time, which is much longer than the typical vibration time scale. And these sets, they obviously are kinds like something like the average geometry of the thing, and in chemistry they are called the main conformation of the molecule. In a mathematical setting, we would say these are metastable sets. So if the system is, or almost invariant sets, if the system is in these sets, then it stays there for a long period of time, long compared to the fastest motions, for a long period of time before it makes more or less sudden jumps to other of these sets. Therefore, in this special case, I have only 3 of these sets. And if I would be able to identify them, then I could, if I'm only interested in long time scales, so I'm not interested in all the tiny details, I'm interested in the real motion of the system on long time scales. Then I could also say, okay, take these sets, use the fact that they that the molecule does sudden jumps between them, and cook up a Markov chain. Fix a time scale and tell me what are the jump probabilities within this time scale between these sets. And then you would get a Markov state, a Markov chain model for the motion of this butane molecule on very long time scales, ignoring all the fastest motions. And surprisingly, this concept works for very many molecular systems. And what does it do effectually? It finds sets in state space in which certain mixing properties, mixing is fast, are there, and other sets are separated from other sets, and the motion from one set to the other is fast. And if the system just has so many of these states, these main conformations, then I in principle am able to cook up a kind of system description of my system which is not so large and has all the jump probabilities between these different states, or the jump rates between these different states. And this would describe what the system is doing on very long time scales, instead of using all the particle-based descriptions where you cannot go to these time scales. And the reason behind it is that the system in this case, in molecular dynamics, if you would look at the energy landscape, then the dynamics going on is something like this. So you have an SDE in this case, a Newtonian motion car here, and then something which is stochastic and damping here. This system is called Langevin, and it has some boundary conditions and things like that. It generates a certain statistics on state space, which is called the invariant measure. And based on this, you can say that there are even very rigorous results on this. If you are somewhere here, then the system stays here for long periods of time, because it's a rare event to cross this barrier and go into one of the other worlds. So the fact that you have these metastable sets comes from the structure of these energy landscapes in these cases. And if the energy landscape has this structure, that there are some main wells in the landscape separated by high barriers or very difficult regions to cross, then you will have metastability in your system, and this Markov state model description will work. And in fact, it seems to be the case that this is very often the case, that the landscape has this structure. And the idea to describe this, the mathematical background, is using transfer operators. So you would look at an operator which is not describing every single particle in the system. It just tells you how functions are moved by the underlying dynamics. So for example, you could write down the probability distribution of all particles in your system and how the probability distribution moves in time. So the evolution of the probability distribution. And a transfer operator is only telling you this. So you give it a probability distribution in the beginning and the jump probabilities of your induced by the dynamics, and then it tells you after some time t what the probability distribution is then. So this kind of operator can be written down for all these systems. And it has certain structures, especially it is a Markov operator, so it preserves certain mathematical quantities. And the spectrum is contained in a unit circle in a complex plane. And if you are ergodic, which is the case in molecular dynamics, then the leading eigenvalue, the largest one, this is the largest one. 1 is the largest eigenvalue. Everything else is smaller. And this is even a simple eigenvalue if there's just one invariant measure. And under these circumstances, you can do a lot. If the system is reversible in addition, then your transfer operator is even self-adjoint, which is satisfied in most of the molecular dynamics descriptions. And then the idea is, okay, my dynamics is nonlinear, stochastic, multi-scale, and I do no longer look at the dynamics itself, I look at the transfer operator, which is living in a very high-dimensional space, but it's a linear operator. So the idea could be, if I have a linear something, then perhaps I can do something about it. And the general idea that is used in this field is this. Let's assume this is my molecular dynamics case, the easiest case which can be considered, where the energy landscape is this V here, and X is the vector of all atoms, or all coordinates of your atoms in your system. Then you would write down the probability distribution, and this would satisfy an evolution equation, which is given by a generator, which can simply be written down as a differential operator. And then you could, if this has some dominant simple eigenvalues, which is the case in very many cases, and eigenvectors associated with them, then you can approximate the full transfer operator by something like this, where these lambda are the ones, in the case of the generator, they are very close to 0. So you have some eigenvalues, simple eigenvalues, which are very close to 0, and all the other ones are far away from 0 and negative. So if you take the ones which are close to 0, then this is a good description of the behavior of your system. So if there are just a few of them, then the long-term behavior of your system is almost perfectly described by these eigenvectors, so by this part of the operator, and everything else is exponentially fast decaying with a relatively high rate. Surprisingly, this is the case even for very complicated molecular systems. And therefore, we can use it in order to approximate the dynamics of a molecular system. So how is this done? The idea is, if you look at it from a scientific computing or numerics perspective, then you would say, okay, what do I want to do? I have a state space, okay, high dimensional, but I don't care at the moment. I just take it and discretize it, for example, into n subsets, boxes. So that is just, you do, if you want to imagine it that way, then it's a uniform discretization. These boxes are non-overlapping, and you get, if you discretize your if you want to know what your dynamics is doing, then you ask the question, if I start in one of these sets, what is the jump probability after some time scale t, tau, into another of these sets? This gives you, obviously, a stochastic matrix. So a Markov chain description of what's going on in state space. And surprisingly, this can be written as a discretization, as a Galerkin discretization of the transfer operator in mathematical terms. If you just take the Galerkin operator of your full transfer operator of the full system onto this ansatz space, then this identity tells you that the discretization matrix will exactly be the jump matrix between these sets. That was the start point of what is nowadays called Markov state modeling. And in a more abstract sense, what you do is a projection to a subspace, in this case this one. If you want to do computations, then the subspace is finite dimensional, and you replace your transfer operator by a projected transfer operator. And this projected transfer operator, like in this case, is a stochastic matrix, so it inherits many of the Markov operator properties of the transfer operator. That's the mathematical background idea. And then you can go to cases like, for example, this. Ask for a peptide which has certain properties. In this case, the construction of it allows you to even observe what you compute in experiments, because you get a laser signal or a light signal or fluorescence signal. If the blue and the red sting are far away from each other, and if they come close, then you no longer get a fluorescence signal. So what the system does can be seen by the fluctuations of the fluorescence signals. This is done in a lab today, and it agrees perfectly with these computations. And in the background, you know, more or less, for this peptide, that perhaps this is just a two-dimensional representation of the energy landscape. It has these 4 deep wells, so dark blue are very low values in energy, lighter colors are higher values in energy. So you see, we have 4 wells here. If the system is there, then it will stay there for long periods of time, wiggling around. And the process of going from here towards there is a rare event, so needs very long timescales to appear. Now, how can I do this discretization of the transfer operator? In the easiest case I can imagine, I simply take state space and do uniform discretization. And then, as I said, if I would start in the box very many trajectories and look at them, I would have the case that all these trajectories together would create this stochastic jump matrix between the boxes of my system. This seems easy, but the questions are obvious. The main problems are, if I do this in a very high-dimensional state space, how can I avoid the explosion of the number of boxes? Obviously not. And the second question is, how accurately really will be the information about the long timescales, the information I really want? I do not want to have all the small timescales and do small time steps, because then for a complex system I can never do it. If you look at the system and you would say, okay, 4 boxes perhaps are enough. If the system stays here for long periods of time, before it does a jump here, it just has to cross this barrier, and then I count jump rates between these 4 boxes, and I get a 4x4 stochastic matrix, which tells me everything about the system on long time scales. In this case, this may be correct, but you don't know what happens here. If the system goes out here, does it directly go there? Or does it perhaps have processes like it goes there, but goes back and then goes there, and so it recrosses this barrier? You don't know. In this special case, a 4-box discretization is sufficient to understand this molecular behavior. But if we are going into more details, then we have to find more structures and invest more mathematics in order to understand it. And there are already books about this, and there are about 1,000 articles doing this already at the moment. So this has become very popular in molecular dynamics. But I want to show you something else which is important for the things I want to do later. You could also do a different thing. You could say, let me imagine that I have just the core sets at the heart of the metastable sets, the bottom of my wells. And I call them C1 to C4 in this case. I don't go and try to uniformly discretize my entire state space. If I would have this, then I could construct something which is called the committers. So if I am somewhere here, then I ask the question, what is the probability of my stochastic dynamics that I started so that the core set which has been touched last is one of these. So if I am here, the committer Q1 is the probability that I came from C1 last, that my trajectory came from C1 last. Obviously, that's the probability. Or it means I can show it in this way. So here, deep blue is values almost 1, deep red is values almost 0. And that's the probability that my dynamics came from this set last. That's not so surprising. I can do it for C2 and for C3 and also for all 4. What do? They are called committer functions, and they obviously are a partition of unity of state space. So I can use them to make a discretization out of it, to use them as an ansatz space for a Galerkin discretization. And if I do this, if I do discretization of the transfer operator based on this committer space, then again I can write this as a certain case of jump matrix. So I again get a stochastic matrix as a discretization matrix back. Or more exactly, I have to replace the eigenvalue problem of the transfer operator by a generalized eigenvalue problems. The details doesn't matter at this point. The important thing is I do not have to do box discretization. I also can use more clever ansatz functions where the dynamics, the structure of the dynamics is already built in. And then a few of them will be sufficient to do the discretization. And again, my ansatz space will define a projection and the discretization matrix will be the projected transfer operator. And then I can write down, it can do mathematics and understand when this is good, and I don't want to go into details, but the main thing is I don't need a Markov property in order to make it a good approximation. So this word, Markov state models, it's a little bit misleading. You don't need that the sets are Markov states of my process. You just need certain approximation properties. Keep this in mind. So what did we do? We have a particle process, molecular dynamics, which is essentially multi-scale, very many timescales. It has these metastable sets. I discretize this process in a very high dimensional setting. I discretize it by just a few sets, which are more or less coming from the fact that the dynamics within these sets is staying there for long periods of time, samples the statistical distribution almost perfectly before it goes on and goes to somewhere else. And by doing this, I get to a rather low dimensional copy of my very complicated system, which still helps me to understand almost everything on long time scales. And nowadays, you can even, if this is the time scale the molecular dynamics itself, the full model really has, and this is the time scale my Markov state model description has, then nowadays there even is an upper bound, an error, a way to understand or estimate the error of this modeling in terms of the time scales. And by this you can guarantee that the timescales you are describing by your finite dimensional copy is a good estimate of the timescales the real system has. Why is this important? Or why are these things interesting? Let's, for example, assume that we have a molecular system, and this already leads me now in the direction of chemical dynamics or biological processes in cells. Let's assume I have the blue structure, which is a larger molecule, or this is part of a larger molecule, and the red structure, which has the effect that it is more or less binds to two parts of the larger molecules. It binds here and it binds there. And then it's a so-called bivalent or multivalent docking case. So there's not only one place in the molecule where the smaller molecule can be attached to, there are two places, or even n places where it can attach. And then what can happen, if I want to understand the reaction of the small molecule and the reaction rate of the small molecule with the larger molecule, and then the small can be unbound from the larger one, it can be attached but just at a single place, or it can be attached at more than a single place. And this is somehow in between, because it could be attached on that side but also on that side. It could be bound on that side or on the other side. So a reaction process itself has very different stages. It can form a complex and is really doubly bound, and the off rate is very, very small. Or there can be these cases in between where it just bound to a certain part. So in terms of the committer, you would have a case, let's say this is the state space where it is doubly bound. This is part of state space where it's completely unbound. And in between you will have the singly bound systems. And in reality, what will happen is that the real binding process will go from unbound into the direction of the fully bound system, but it will more or less often enough cross this level where just one bound is established. So it will bound to the system with one end, then get free again, bound with the other end, and do this many, many times. This is called rebinding, before it is really attached. And then if we think in the direction of the systems which have been explained this morning, where we have descriptions of molecules which can have reactions between each other in a certain ball in state space, so they can only react if they come close enough or not, then this obviously is something which is of importance, because whether your rate that you put into the system is correct or not depend on whether the system is doubly bound, singly bound, or whether these structures in between are. So the process of rebinding is something which is dangerous if you're doing it wrong. But we are able with these Markov state models to more or less cook up a very small model for this with some discrete states. In this case, for example, doubly bound and unbound, or even doubly bound, single bound and unbound, and have all rates between them based on molecular dynamics descriptions. And this system has the form of a chemical master equation. It's just a jump process between states. It can be more or less clicked into a chemical master equation for biological processes without changing anything. So this way of looking on molecular reaction processes is more or less perfect for doing stochastic modeling of biological reaction kinetics in cells. And it can even handle the difficult cases. So for example, this is one of the publications which came out just this year. There what you see is a large protein, different conformations of it. They don't look so different from just looking at them, but if you go into the details and look how the ligand is bound or unbound, then it's really about multivalent binding. And we already know that, for example, in this case, whether it is bounded in this part or in another part is very important for the biology going on in the cell, because one part, especially this one here, activates certain cellular processes and the other parts don't. So it's very important to get this insight into these details. And if you have it already on a rate level, then you can more or less immediately use it into and plug it into your cellular simulations. Imagine that you have viruses or something like this, or more complex particles in your cell, which also happens often enough on membranes, on scaffolding and whatever. In this case, it is also a kind of multivalency or rebinding, because there are lots of tinier molecules which can bind to this, but they can also, lots of them can bind to this, and then, for example, it can no longer attach to a surface, or it can. So all these processes are very detailed, and if you understand the rates carefully enough and distinguish between the different modes of being bound or partially bound or unbound, you can have much more detailed and much more accurate descriptions of the dynamics going on. But what does this mean for particle-based reaction-diffusion kinetics, as they have been explained this morning? So, as we learned or have seen this morning, there are many of these particle-based reaction-diffusion schemes where every particle is described as something which is diffusing around, and then if they come close enough, they can react with each other. And there is a certain parameter in many of these systems which tells you what close enough means. You need this parameter, but normally you are far away or you're close enough, and then you do reactions. So they are all based on this fundamental idea. At the moment, it is possible to distinguish between I am close but still not fully bound, and I'm close and I'm fully bound and I do a reaction or something like this. But you would have to model it directly into your system. But let's imagine that we can do this, that we have a description which is biologically incorporating all events which are important for my system. For example, based on molecular dynamics simulations, which I did before. Then I would have something whose principle is shown here. It's just this reaction diffusion systems. All particles are moving around, and there are just these very simple reactions appearing. And obviously what you see is like a system where blue can eat red and red can eat blue, and so they are more or less changing and forming interesting structures in this limited space they have here at the moment in our cell. And this is what reaction diffusion systems would look like if reaction is relatively fast compared to diffusion, because that's obviously the case. I have very fast reactions here, but the system is at a point so the movement in the diffusion constant is not that large in the system, so it's not well mixed. But these things can happen. But the question is, if imagine that this is happening in a cell, okay, with let's say 10 million particles, you have many different reactions. You can imagine that, and this is the case in all of these typical algorithms, that can be, from a computational viewpoint, can be very complex. So timescales are very small. The time between two reactions can be very small, which means that your algorithm, even if you use tau leaping or other tricks, has to make rather very, very small time steps, which means in order to go to longer time scales which are biologically interesting, you need to do, your time stepper needs to do very, very many time steps. And what you see here is just one stochastic realization of the process. So in order to get expectation values, you would have to do very, very many of these simulations. And each single one is very, very long in time, has to do very many time steps. So computational, we are in a similar situation with these particle-based simulations than we have been in molecular dynamics. We would like to go onto timescales which are long enough to be biologically interesting, but the timescales of the inherent processes in my system force us to do rather small time steps. So how can we more or less go on? And the way which has been discussed this morning is about reaction-diffusion master equations. So the idea is, don't let every particle move around in your space and do your reactions, because if you have 10 million, then that's induces a lot of computational problems. Induce a grid discretization of your computation space, in this case of your cell. Make it a fine discretization, so you will have many cells in which things can happen. But know that the things which are important are the cells. It's not longer the number of particles you have to look at. And then you get a discretized description of the process, which has the structure and the properties which have been discussed this morning. So I don't have to go into details about that. But let's discuss what is going to happen. If the grid size is becoming small, then perhaps in a grid, if you choose the right grid size, you do not have so many reactions happening. But still, the jump between the grid size will be fast, the jump process itself, organized or induced by the diffusion. So whatever you do will always have small time steps in your system. If you have very fast reactions and they appear everywhere, then you also will have very small time steps. So it can be a very, very complicated process based on the discretization of your cell. So it's a way to go, but perhaps it's not, and that's the case at the moment. It is very difficult to do this in all details. So what we propose is something which is combining the stuff I have explained to you before and this idea of replacing reaction diffusion particle-based simulations by chemical master equation with a certain spatial component. So the idea I explained before in molecular dynamics is you have particle dynamics, and if you are able to find certain sets in state space in which mixing is fast, and may it be even for certain parts of the system, then you can use this as larger discretization sets. So you can, by looking at the structure of your diffusion process, you can cook up interesting discretizations for state space, which are leading to much larger, coarser discretization sets than the fine discretization you would normally do. If your process has this property, then it is clever to use this property and just use it as a discretization in a very coarse sense. Okay, what you will have to have is fast enough mixing within these sets, but then you get a much coarser description of your system, which is not only an advantage because your discretization is coarser, it's also a main advantage because time steps will be larger. And then you can go to longer time scales, the time scales you're interested in. So this idea would formally, if you want to write it down, mean that within each of my subsets, I would assume that my diffusion is fast. I would assume that the transition between my subsets are rare, and that in each of the compartments the reaction rates are more or less constant, independent of the position. These are strong assumptions. And please keep in mind, like in molecular dynamics, I do not want to more or less take my cell and decide before everything else which kind of discretization sets I want to use. What I want to do computations and find out about the discretizations which satisfy these assumptions, and then use them for discretization, and then would allow me to go to longer time scales than I would be able to do with just the fine discretizations. That's the philosophy. So I'm not saying that this is almost everywhere satisfied or something like this. I have to be very careful and have to base this on data I get from simulations in order to find the right sets, in order to allow my simulation to go to longer time scales than I was able before. So in order to do this, assume that I have sets like this, then sorry, I would again, for the spatial part, I would get a Markov jump process with a rate matrix, and this rate matrix would be built out of rates that I get from more or less looking at my diffusion process and its jump rates between these sets, which are now, because the sets are assumed to be metastable, these jump rates are much smaller than the jump rates between all these fine sets, because I ignore the fast timescales I'm not interested in. And then I could go to diffusion and reaction and could write down my state space in the same way I did it for, or somewhere I did it for reaction diffusion master equations. And then I would have a state which is can be a very large matrix or a larger matrix. And by writing everything down, jumps, diffusions, I would have again a master equation which now is containing reactions and jumps between discretization cells like before, but now the discretization cells are larger things which are designed by already looking at the process for short periods of time, which satisfy my assumptions, so that the dynamics of this object allows me in a numerical realization to do much larger time steps and have much less states in the system than if I really would do fine grid discretization of everything. So if I would look at this, and I'm a computation person, from the aspect of computational effort and accuracy, then perhaps the best we can do is full particle-based reaction diffusion things. This is very accurate if you have all the rates you need, and if you can design the right parameters, which is sometimes very difficult. Then you have reaction diffusion master equations, which is doing fine grid discretization. It still has very small time steps, but doesn't suffer from the fact that perhaps you have very, very many particles in the system. Then this is the spatial temporal chemical master equation system we would prefer, where you discretize your state space, your spatial regime with larger sets which satisfy certain properties. So the time scales, the time steps are longer, and the spatial resolution is less. So it's not as accurate, but if you do everything correct, it is much more efficient in terms of computations, and then all the other descriptions down to the boring, simple reaction diffusion case in terms of differential equations. So, and now if you want to solve a problem, then you have to find your way. You have to answer, which is the computational effort I can really invest on the biggest computer I can have? Do I have all the rates needed for that? Or do I, in order to achieve the timescale resolution I really want, so to go on long enough timescales for my biological processes, do I have to reduce my computational effort? And if I have, what is the right description in between? So let's just go for a last point in this talk in order to explain something as a kind of illustration and about things that can happen. Let's assume we just have a nucleus and a cytoplasm. And let's assume that in between there's a membrane. Okay, it's highly idealized, but I just want to explain something to you based on this example. In this case, for example, if the membrane doesn't let everything immediately through, so there is a certain membrane permeability, then it means that the nucleus, for example, really is a metastable set in the sense I explained, because the dynamics, in order to leave it, needs a certain time which is much longer than all the details of the motion in between, as long as the reactions which happen in the nucleus are faster than this process. The same can happen here then. So we have our two sets in this special case, depending on the permeability of the membrane. So if it is large, then there will be metastability. If it is small, then perhaps there won't be metastability. And then you can look just at the diffusion process, ignoring all the reactions, and can ask what are the jump rates of the diffusion process in this setting. And you can cook up such a Markov state model description for something like this. Okay, this is in a very simple case, just reflecting what you would have to do in a cellular surroundings for every species in some sense. But you would come up with certain rates. And then, if you also have reactions, you can more or less also get the information how the reaction timescales more or less depend on the radius of the nucleus and the radius of everything. So this can be studied just based on data coming from simulations. You don't need analytical expressions for this. You just start the processes, get data from your thing, and try to fit something which is a Markovian jump process to that. And this is very fast because you don't have the full system with all the reactions, and you only need short times. So at the end, you get a certain reaction rate constant, which then appears in the system in the propensity function here, if we have just the directions I told you. But now let's take a system in which I have two reactions, these two here. And in the first case, they are both happening in both compartments. So now I know the nucleus and the cytoplasm is no longer a discretization set in a certain sense. It is a discretization set from an object, but it's also a kind of compartment in a biological sense. But what can happen now? Let's go back to our cycle. What can happen is that a particle, more or less, is here. It leaves the nucleus. If the permeability is not too high, then this process of leaving is fast. It goes out. It can do rebinding there and then come immediately back. So you have these recrossing events. The system can go out, back, out, back, out, back. And then it's difficult to distinguish where the reactions happen. So the same problem then in the molecular system appear here also, because you put boundaries to your system. And at these boundaries you can get problems from the process crossing the boundary and immediately coming back. And these problems lead to the fact that not always your approximation properties are good, even if you did everything according to the Markov jump properties correctly enough, and even if the mixing is good enough. So if you want to do something like this, then for example, you can have the case where this process is not allowed to happen in the cytoplasm, but it happens in the nucleus. If this is the case, then you see that immediately our process is a much better description of everything. Or if the reactions are separated, so one takes place in one component, the other in the other end, there is a huge enough permeability so that you really have mixing, so you really have metastability in your sets. What I tell you here is that you have to be careful about your assumptions if you do something like this, that a process like rebinding is important and has to be modeled on a molecular level already, carefully enough, incorporated into your rates, otherwise you can fail. And the other thing that I want to show is that that you can get it done if you are careful enough. So let's repeat the philosophy. What I want to do is I want to have a cellular reaction process. The first thing I want to do is, for all the processes where I don't know rates or which are a little bit more difficult for the things I want to explain or observe, I perhaps have to do molecular dynamic stuff before. If I do this correctly enough nowadays, it's possible to immediately get the description which is discrete in the sense of jump rates between conformations, or this means in the chemical sense you immediately get the reaction rates. Even for complicated processes like rebinding or ligand binding or trimer or other things forming, aggregation, you can have detailed descriptions of what's really going on. So if you do something like this, you get a good description of the rate in your system. And then, if you do not want to do stochastical-based particle reaction diffusion systems because it's too complicated and you cannot go to the timescales you're interested in, then in my language you have to coarse grain. You have to introduce discretizations. The first step of discretization you can do, you introduce a uniform mesh in your cell, then you get this reaction diffusion master equations. They have the disadvantage that you still can have very small timescales and very many discretization sets for very many particles. And then if you want to further coarse grain, then you use the same technology which has been developed for particle-based systems in molecular dynamics. You can use it again in order to form coarser discretization sets and compute the jump probabilities between these just based on simulations and on data, not on analytical calculations, which allows you to coarse grain your description, and then your time steps will be longer and you have less discretization sets, which allows you to go to longer time scales for your simulation. And that more or less is a cure for at least some of the problems which are associated with multiple time scales in your simulations and where you cannot go to the biological time scales of interest or to a complexity of the system of interest. And from my point of view, nowadays this is a field in which all these multi-scale things have to be combined in order to get really reasonable and reliable and accurate descriptions of the processes that are really going on. And the more of these tools we have available in standard software, the more easier it will become in order to really understand what we are doing in these kinds of process simulations. These things, for example, this Markov state finding for objects like this, they are available already in software that you can have, for example, from our website, but also from others. It has been designed for molecular dynamics, but it has now been adapted to these cases. And there is this software for particle diffusions ready, and it's already an interface for this. So these things are available, and everyone who wants to play around with this and see what this would mean for his or her system is invited to do it. And as a last remark, let me make a mathematical comment. Like for molecular dynamics, there is this error indicator. I showed you the slide. So you can really have an error indicator for the accuracy of your Markov state modeling. So for the accuracy of your discretization sets. It's not fishing in the dark. If you invest enough simulations to really do this nicely and reduce this error in a nice enough way, you will get very accurate descriptions without an enormous computational effort. This is what I wanted to tell you. Thank you very much for your attention. Let me perhaps point out, Steffi, I already told her. Frank is the one who does many of the Markov state modeling stuffs in molecular dynamics for ligand-protein and ligand protein-protein association processes. Marcos is doing multivalency systems and rebinding, and he's an expert in this. Eric is a colleague from NYU, with whom I worked in this topic, Markov state modeling, for a decade now. And Carsten Hartmann is someone who is really understanding more of the mathematical stochastic dynamics details in the background. Thank you very much for your attention.
I have a couple questions.
Please, yes. So I would, for the spatial temporal chemical master equation, it seems that you need to know in advance like the energy landscape, or at least have some knowledge about it. And
no.
You don't?
No.
Okay, so but then
what we have to do is we start simulations of the system, and from the simulations they produce data. The right analysis of this data tells you which are the sets that you should use for which species.
But wouldn't you enable to see like the rare events would need like data that runs for a really long period of time?
Sorry?
So wouldn't you need
no. That's the point. So I cannot guarantee this for every system. That's obvious. But if I start my simulation of the fusion processes, even including the simulations for lots of short trajectories, Yeah? This is what I can do on today's computer architectures very nicely. So start very many parallel trajectories of your system. Then if my error indicators decay, I find a discretization which is rough, but still on very long time scales has a very good description of the underlying processes. This discretization I would take without knowing it in advance, just based on the data my simulations have produced, and then I would do simulations on very long time scales based on the coarse-grained master equation which comes out of that.
Maybe a similar question. So, from a biophysical point of view, if you look at proteins, the fact that this method works, I think, is related to the fact that proteins have these nice energy landscapes with nice minima and barriers in between. Is there a reason, if you think about a cellular reaction system, that the energy landscape would have such a property? Otherwise, I think you might start with some rough discretization, but the coarse graining would not get very much coarser than that.
So two answers to this. The first answer is, in some sense, it depends. If, for example, you would like to look at a cell, if there is a membrane, you could look at this as a peak in the energy landscape, or as just as a membrane, as a boundary somehow. So the perspective is different. But in general, the only thing I can say, you can do this, you can start these algorithms, and if you get a description, if you find a discretization for where the errors can be reduced to a certain extent, then you can take it. Again, I cannot tell you that you can do this for all possible systems, but I can tell you that you can do it for a lot of systems, not for all. And what kind of spatial objects come out of this depend on the question you want to ask. So what is really in your system? Sometimes something very crude like nucleus and cytoplasm is enough. For other questions, you need much more detailed sets and then more of them. But the essential thing is that at the end, the aim is to allow longer time steps. That's more important than reducing the number of discretization sets, in my opinion. And based on the experience we have made so far with this and with molecular dynamics, we did it for almost all cases. For example, in molecular dynamics, it's done for DNA, RNA, proteins, peptides, even for completely different things like nanotechnical systems, everywhere. Even where you don't have an energy barrier, if there is a diffusive part between these two core sets, then with this committer space analysis, even this can be handled. So you don't need a barrier. You just need a dynamical time scale of mixing in one set and in the other set, and then the right kind of discretization. So from the experience, we can only say it works in much more systems than we would normally assume. But the only guarantee you have is start it, look at the error. If you get it small enough, take it.
Okay, any last questions? Yeah, please.
Sorry, if you use, if you have a multi-scale problem, if you use, for example, some implicit methods, related to the tau leaping, this can not help, your method. This will not, reduce the computational cost of your method.
You know, we have used tau leaping for very many cases with non-spatial master equations. And even there, you have very many cases where you cannot reduce the time step if you want to be accurate enough, simply because you have such a mixture of time scales. There's not just one fast process, and then you can do tau leaping because more or less this fast process is averaged out in the right way. If there is a real spectrum of time scales, then it can fail in many different ways. But at the moment, we did it for spatial master equations. And I have to admit that we never really were happy with the results in tau leaping.
Even using implicit, schemes?
Even using implicit schemes. If there is a spread of timescales, which is rich enough. So again, for systems where I have some fast timescales and then some slow timescales, everything works. But if I have more or less everything in between, I get so many problems that I cannot handle it.
All right, so
My experience, not a final answer.
All right, so we'll hold further questions and discussion for the break. So let's thank Christof one more time.